So this is the third video of the chapter Biotechnology and its applications. In this video, we will discuss some more things about endonucleases, how it cuts the fragments, okay, and what should be the qualities, the characteristics of a vector, okay. So first, moving on to the endonuclease. At first, we have to learn one thing that is known as palindromic sequence. Now, palindromic sequences are the sequences that are the same when they read from front to back or back to front. For example, this word, if you read it from front to back and back to front, it will sound the same. The meaning does not changes. This is the palindromic sequence. This is known as palindromic words. Okay. So, for example, case 1. Okay. There is this sequence, and in the case 2, there is this sequence. Remember, in the both the sequences, if you notice, if you read it from 5 dash to 3 dash and 5 dash to 3 dash, it remains the same. Okay. So if you read it from front to back or back to front, it is same. Similarly, front to back or back to front, the sequence remains same. Now, another important thing is that this bases are connected with one another by phosphodiester bond. Similarly, these are also bounded by one another by phosphodiester bond, right? And I am marking it with a black pen. These are bounded by hydrogen bonding. A is bounded by thymine by 2 hydrogen bonding, okay, and G is bounded by cytosine guanine by 3 hydrogen bonding. Similarly, here also, right? These are hydrogen bonds. The black color represents the hydrogen bonds and the blue color represents the phosphodiester bond. Now, for example, for the endonuclease, okay, the restriction site is GC, so it will cut here. It will cut here, right? So, for example, the restriction site here is here. It is GC, and here, for example, it is AT, right? So here it will attack, and here it will attack. Whereas it will the endonuclease will attack here and here. So after the attack, what will be the product? What will be the result? Right? Let's see. Here in the second case, the result will look something like this. 5 dash. Plus. Right? And see the hydrogen bondings between this A and this T, this A, this T, this T, this A, and this T, this A. It has been broken down. There is a chance that it will again regain. It will again make these hydrogen bonds. So it will stick together. So it is also known as sticky ends or cos sites. Okay. Sorry. Whereas, let's consider this part. If we see the product, it will look something like this. See, here the phosphodiester has been broken. Here hydrogen bond. Here hydrogen bond plus phosphodiester bond broke. Whereas here only phosphodiester bond broke. So there is no hydrogen bonds left, right? Right. There is every hydrogen bond intact, right? There is no hydrogen bond left that needs to bond with each another to regain its original conformation or something like this. So these will not stick with one another. So it is also known as blunt end. Okay. So this was two cases, how the endonucleases can cut and how the products can be different. Now, we'll learn some more things about vectors. What should be the characteristics of a vector? We'll learn about vector. Vectors, characteristics. So the number one characteristic of a vector is it should be small in size and it should have large gene carrying capacity. And this gene carrying capacity is measured in kb. Kb means kilo bases. For example, you have 20,000 bases. That denotes that you have 20 kb gene. Okay, a gene if consists of 20,000 bases, it implies that it can be called that you have 20 kilobase of a gene fragment. Okay, now it should be able to originate, replicate on its own. So it should have a ORI site. Third, it should have restriction sites so that you can cut and make your own manipulations. So it should have restriction sites. Okay. Now, the other thing is that it should have ROP protein. ROP. ROP is also known as protein or for or of replication. Okay. Protein of replication or protein for replication, you can say. Okay. And it should have another thing, selectable marker genes. Selectable marker genes can be antibiotic resistant gene like ampicillin resistant gene, kanamycin resistant gene, etc. It might be any gene which produces an enzyme, example LAC-Z, that produces beta galactosidase. Okay, let me write it down for you clearly. Okay, you have read it in ERP operon. Sorry, lack operon. Lack operon, you have read it, right? So these are the characteristics that needs to be there in a vector. Okay, one is low size. Okay, so that you can easily manipulate with it, you can work with it. The more the size of a vector, the more complex it will get. More gene carrying capacity. For example, your insert consists of 40 kilobase. Okay, and the vector can only carry 20 kilobases of insert. So you won't be able to use that, right? So high gene carrying capacity, ORI site, because it needs to replicate in the host body. There should be restriction sites so that cuts can be made. There should be ROP. Okay, this ROP, it binds with the ORI site. Okay, and this complex is recognized by DNA polymerase. Okay, it is recognized by DNA polymerase, and the DNA polymerase binds here and starts its replication by this process. Okay, so this is the reason the replication of protein or replication or protein for protein for replication is needed. Okay, now it should have a selectable marker gene, either antibiotic resistant gene like kanamycin resistant, ampicillin resistant, or it should have gene that produces enzyme, for example lactoferrin beta galactosidase. In the next two videos, after two videos we will discuss about this blue fight screening effect and how is it done and what is not, what is there and what is not. Okay, now let's understand some few things about plastids. Sorry, plasmids. Okay, this is not plastid, this is plasmid. Plasmid, you can say, is, for example, this is a bacterial cell. It has its own genetic material. This is its chromosomal DNA. Apart from this chromosomal DNA, it might have some extra chromosomal DNA. Okay. Now, this extra chromosomal DNA is double-stranded, covalently closed, circular, and it might have antibiotic resistant gene. Okay. Up till here, let's end this video. In the next videos, we will discuss further about these things. Okay.